Let's begin with the male reproductive system. It consists of the gonads or testes, as well as accessory organs. Accessory organs include the copulatory organ or penis, the glands, which secrete substances that become part of the semen, ducts, and tubules. The testes are housed outside the abdominal cavity within a sac called the scrotum. The scrotum consists of an outer skin layer, a middle superficial fascia, and an inner layer of dartus muscle. The dartus muscle, when it contracts, wrinkles the skin of the scrotum. The testis is shown here in white in this illustration. It is surrounded by a serous membrane called the tunica vaginalis. As with all serous membranes, the tunica vaginalis consists of an outer parietal layer and an inner visceral layer. Sandwiched in between those two layers is a cavity filled with serous fluid. Deep to this and surrounding the testes is a fibrous capsule called the tunica albuginea. The tunica albuginea moves inward to create septa or walls that separate the testes into 250 wedge-like structures or compartments called lobules. Each lobule contains up to 4 seminiferous tubules. The seminiferous tubules are the site for sperm production. Once sperm are produced, they will eventually make their way into the epididymis. The epididymis consists of about 20 feet of ducts that are convoluted or twisted. Within the epididymis, the sperm mature and gain the ability to swim. They are also stored in the epididymis until ejaculation and can be stored for several months. It takes about 20 days for the sperm to make their way through the epididymis. This gives them enough time for the maturation process. If ejaculation occurs, the sperm move into the ductus deferens or vas deferens. We'll continue to follow the sperm later. The testes are supplied by the testicular arteries, which are the gonadal arteries. They branch off the abdominal aorta. They are drained by a plexus of veins called the pampiniform plexus. I'll mention these again later. This is an 11-month-old toddler. He has an inflammation of the scrotum, which is due to an accumulation of fluid within the cavity of the tunica vaginalis. If the fluid is serous fluid, the condition is called a hydrocele. However, if it is blood, the condition is called a hematocele. Transillumination is used to determine whether the patient has a hydrocele or a hematocele. This involves taking a light and shining it through the scrotum. If the light can be seen on the other side of the scrotum, the patient has a hydrocele. This is due to the fact that water allows light to go through it much more easily than blood does. Why house the testes outside the abdominal cavity within the scrotum, where they are vulnerable to trauma? The answer to this question is temperature. Sperm production requires a temperature that is 2 to 3 degrees Celsius lower than internal body temperature, which is 37 degrees Celsius. The testicular arteries supply the testes, and the pampiniform venous plexus drains the testes. Let's look at how these blood vessels are involved in cooling the temperature or lowering the temperature surrounding the seminiferous tubules. Blood in the testicular arteries leaving the abdominal cavity will have a temperature of 37 degrees Celsius. Blood in the pampiniform plexus draining the testes will have a lower temperature. This will allow heat to move from the testicular artery into the pampiniform plexus, increasing the temperature of the pampiniform plexus back to the 37 degrees Celsius. As this heat is being removed, the temperature in the blood will decrease, as mentioned before, to 2 to 3 degrees lower than the 37 degrees. Varicocele is a condition that occurs in about 15% of males. It usually appears between the ages of 15 to 25 and involves the pampiniform plexus. Recall that veins have valves to prevent backflow of the blood. In varicocele, the valves are faulty, similar to varicose veins. This allows blood to move backwards and pool, dilating the veins. This may impede the ability of the veins to lower the temperature of the arterial blood. Recall that sperm production requires a temperature that is 2 to 3 degrees lower than the 37 degrees Celsius. Surgery can usually correct the problem. Recall that the seminiferous tubules are your sperm-producing factories. They contain spermatogonia, which are your stem cells. In this illustration, these are the spermatogonia. A stem cell is a cell that will undergo cell division to give rise to other cells. The type of cell division that will give rise to the sperm is called meiosis. We'll look at this in greater detail when we study spermatogenesis. Another type of cell found within the seminiferous tubules are the substantacular or Sertoli cells. These are the orange cells, and notice they are large cells. They have several important functions. One is they form the blood-testis barrier. Here you can see some processes that are extensions of the cells. And those processes are bound to one another or connected to one another by tight junctions. This prevents immune cells from having access to the cells that are undergoing meiosis. This is important because when cells undergo meiosis, their genetic information is different than the spermatogonia. They will be seen as foreign cells. The Sertoli cells also release androgen-binding proteins in response to FSH. ABP binds with testosterone, concentrating the testosterone in the area where meiosis is occurring. In addition, they release inhibin, which inhibits sperm production. When sperm count numbers increase, inhibin is released. This helps maintain sperm count. In between the seminiferous tubules is a third type of cell called interstitial cells, and they release testosterone in response to LH.